Welcome and thanks everyone for joining us for our second NRM Regions Australia Biosecurity Webinar for this year. Firstly, can I acknowledge the traditional custodians of the various lands that people are joining us from. I am fortunate enough to be home in Bibbulmun and Noongar Budjar on the south coast of Western Australia. Invite you if you'd like to add your place of the custodianship of the place that you come from in the chat. Can I acknowledge Saj and Trish for all of their hard work for today in advance. They're here dutifully assisting us with various elements of tech and questions as we go through. And also thanks to all of the NRM people that have joined us, and also thanks for the DAFF attendees that have shown a bit of interest and have joined us today. So we really appreciate you coming along. So a few bits of housekeeping. As you would have heard, the session is being recorded. We have been breaking these down into short video series to go online. So that is happening. Can you please mute yourselves whilst you're not talking? Carlos, if you've got a question for Trish or Susan, can you just maybe put it in the chat to them if they can address it? That would be great. I just noticed you got your hand up. So if you do have the chance, okay, thank you, Carlos. If you do have the opportunity to indicate where you are in your handle on your name, that would be great, so we can identify who you are and where you're from. Can you please use the chat for questions, and we'll try to address some of them as we go through, and we'll also carry them through to the Q&A session at the end. So look, just a really brief overview of the topic. Look, whilst we've been talking about this for quite a lot of time, the NRM sector, more now than ever, has been asked to do more with less. And as you're all aware, feral animals are part of the tripartite of fragmentation and fire as really key drivers of threatened species conservation. And so we're really always keen to look at great solutions to dealing with feral plants and animals and invasive species. So today's webinar is going to introduce you to a range of innovative tech to control and identify pests and weeds. And we have 4 great experts from across Australia, from a range of backgrounds and a range of focal areas. And what we're really hoping that you can do is draw some inspiration from the presentations about what you're doing on the ground and how you might be able to make it, apply it to your work to make it more efficient and efficacious. So at the end of the 4 presentations, we'll have some Q&A. So we'll hold all the Q&A to the end of the session, and really keen to explore at the end of the presentations about what's working for you, what else you might have an interest in, what you might see as your key needs and constraints using technology, and how we can really help you transition into using more innovative and technological solutions to the work that you're doing. So with that preamble, I'm going to move into our first presentation. So we're going to hear from Carlos Babativa Rodriguez. So Carlos is a project officer at DPIRD, Western Australia, and he's got a background in computer science and mechatronic and software engineering, which is fantastic. So Carlos has been working in crop protection and invasive species since 2017, and working on implementing systems for capturing and analysing imagery and other data that's integrated with weather patterns, GIS and workflows and other technologies. So this work has included in-crop and in-harvest pest monitoring, the use of microwaves for managing seed beds and bulbs, and the compilation of AI-ready data sets, which amongst other things have been used to detect skeleton weed and rubber vine drones. And so Carlos is going to talk to us about these AI-ready data sets and proof of concept weed detection using drones on skeleton weed and rubber vine. So Carlos, over to you, please. Oh, and you're on mute, mate.
Thanks, Justin, for the introduction. And yeah, I'm just sharing my screen now so you can see. Can you all see my slides?
Yes, thanks. That looks great. Thanks, Carlos.
Perfect, thank you. So yeah, so I'm going to talk about mainly the AI ready datasets that we're preparing for detection of weeds in the landscape. This started for skeleton weed in the wheat belt, and it has now go across to rubber vine. And I'm also going to present a little insight into tap into some work that we've been starting to do for innovative invasive surveillance using pollen and eDNA, two separate components, but that also fit into one another. So basically, skeleton weed has been under eradication in WA for a large number of years, and in 2020, the last review has revealed that more surveillance needs to be done to be able to properly delimit the extent of the weed, and to be able to utilize better the budget. And the cost benefit of the program is 8.6 million over the next 30 years. So, and this is the current extent of skeleton weed. It's being contained at the moment, or the spread of it is being contained at the moment. But more efficient surveillance is going to help a lot with the extirpation of the weed in the areas where it's not widespread. So the typical motivation for eradicating or for using drones for weed detection is it's almost like a holy grail, isn't it? So the expectation is that you will cover vast areas. There's no movement restriction. There's sharp eagle view of the landscape, and you can spot every last weed. But the reality is that there are several constraints in terms of the biology of the plants. Sometimes they only flower for a certain time of day or for a certain time of the year. And the flight and capture parameters do limit your capacity to be able to cover the area that you need to cover during the time of the day that you need to fly. So basically, what AI, how AI works for drone imagery is it does require each image to be 1 megapixel during training. So the imagery has to be tiled up into smaller sections so that it can be fed into the model. And this imagery will be having annotated data. That is whether it is presence or absence of weeds in particular tiles, or weeds marked with squares, or with tight boundaries around them. Each method obviously takes more or less effort, but also it translates into more or less accuracy. We split the data set into a test data set that is set aside for testing later, and the training and validation data remaining is used then. So we put the 80% of training data through the model. That gets tested on a validation data set, and that gets repeated several times, usually 300, 400 times. And the errors start coming down, and then the last set of data that was set aside is tested so that we can get metrics. We can then track the performance of the models. So that's what we refer to as AI-ready datasets that are labeled, that are cut into tiles, and then split into train and test validation datasets. But how do we get there? So it's very important to know where your weeds are and where your images are. So we basically map the footprint of each image, and we map the footprint of each of the plants or clusters of plants that are there. And then we overlay the ground truth data and imagery, and the ground and the aerial imagery. So 2 case studies that we have been working on, skeleton weed and rubber vine. we take a bottom-up approach where we start designing the parameters of the flight based on the biology of the plant. So for rubber vine, we use the flowers that are on top of the canopies. For skeleton weed, we use the stems mainly that are at the ground level. So ground sampling distance or target sampling distance, that will be the factor that we go off to start planning our flights. Skeleton weed has 2 to 4 millimeter branches, and they're very, very, very thin. And the flowers only flash in the morning, so we can't rely on the flowers to detect the skeleton weed. As opposed to rubber vine, where it does have flowers, they're very big, and we can rely on the flowers to detect them. The smallest adequate size for an AI to detect small objects is about 2 to 8 pixels a side, and if we use something that's typical in the agriculture sector, something like 40 millimeters per pixel, we're gonna completely miss the plants. So we need to go with much finer ground sampling distance, 2.7 millimeters per pixel we use for skeleton weed. And for rubber vine, we do use 5 millimeters per pixel. That covers about 6 centimeters would become 12 pixels across a flower. So that's a very good representation of the plant. So for ground truthing, again, we can use for rubber vine, we can use coarse accuracy location of the plants, because the bigger plants we can sort of only know exactly where each of them are. For skeleton weed, we do use an RTK system that gives us more centimeter precision of the position of each plant, so that then we can locate each of the plants in there and put a square around it. We use a GIS processing method that then allows us to ignore everything that hasn't been ground truthed, and everything that has been ground truthed it will be included in the data set. And if there are overlapping images, the tiles are not going to be repeated, or they're not going to be used twice, or it deduplicates basically the imagery. And it also allows us to use visibility scores of the plants. If there are plants that are not visible, that are going to be detrimental for the model, we can also remove them in this process. And then the last step is to go and validate the detections that the model provides us. And this map on the left-hand side shows us plants that have been mapped in swords. Then we can go and have a look at those specific images where the swords have been mapped by a ground officer, by a field officer, and we can run the detection on them and validate the detection to put more data into the model. So this is where it's really important to have humans in the loop, so subject matter experts, biosecurity field officers that are experts in identifying the plants from lookalikes into the system. We use Label Studio. It's an open source software that's very flexible for anyone to use. And we basically train the model, perform some detections, and then we have a validation guidelines for the field officers that use the infrastructure of DPIRD to both locate the plants in the ground with their tablets, and also go back into the office and have a look at what detections we're going to reutilize in the data set to keep on feeding the model either false negatives, plants that have been missed by detection models, or false positives that have high confidence. So this is the main part that I wanted you to be interested in, is using the human in the loop. Currently, we are detecting skeleton weed plants. Even though they're very thin, we can detect them in the landscape. And this photo on the right, it's just one photo of rubber vine that was taken from a helicopter. It was using a different camera from a different height. And even then, the initial model was able to pick up all of the flowers that are visible to the human eye. That was a very satisfactory result. So we're still building the models for it, and we have started tracking the progress of each of the models. So we do want to take that, recall that sensitivity metric, drive it higher and higher upwards. And the only thing I wanted to present about pollen and eDNA, there are initiatives that we have started looking into. Also, that will be working on for surveillance of species in the bigger landscape. So the wheat belt is a reasonably big area, up in Kimberley, recently big areas where we can't always go and have a look at plants ourselves. So active surveillance is very challenging. So passive surveillance methods like pollen traps, we've started to look at. So this is actually an automated pollen trap that our colleagues in grains have developed. It has a carousel that changes every couple of days. You can have 8 different slides with glue to capture pollen, and it's being used for monitoring rust spores in the wheat belt. So we reutilize them in summer when they're not utilizing them. We reutilize them to monitor for skeleton weed, and this is the reference imagery of what the skeleton weed pollen would look like under scanning electron microscopy. And we have some imagery that we have captured by scanning the slides, and both using pollen and the pappus of the seed, of skeleton weed seed, we can get some passive surveillance occurring. We have had challenges. So when we use the glue that is being used for rust identification, it does it comes up with too much noise for the scanned slides to be able to be useful for training an AI. So we have been exploring alternatives like positively charged slides, which don't require any glue. They simply attach to the surface of the slide by the electrostatic of the slide. The other surveillance that we have started looking into, and CSIRO is doing some sensitivity tests for the primers that are being developed for skeleton weed, is eDNA surveillance. So we this is an oversimplified view of what the system would look like, but we basically want a system where we can go out in the field and survey or sample either troughs or dams where the skeleton weed has been eaten by sheep or kangaroos, or the wildlife, and we will have an in-field quick PCR system that allows us to verify in the field whether the skeleton weed has been present or absent. And we are looking at other alternatives like feces to be able to do this. So this is still in its early stages, but yeah, very promising technology that could be of use for many applications. And that's it. So I want to acknowledge the industry funding scheme, Grain Seeds and Hay, who has funded the project for skeleton weed research, and everyone involved in the process. Please get in touch if you would like to discuss. Yeah.
Thanks, Carlos. That's fantastic. Two for the price of one there. So that's excellent. There are a few questions in the chat which you might like to attend to, or we can carry to the end. We're going to hand over now to Jane Kelly. So Jane's a senior lecturer in livestock production management at Sturt University. and a researcher within Charles Sturt's Gulbati Institute. Jane's most recent experience explored the opportunities and limitations of remote sensing for weed detection in a complex landscape. Her and her colleagues have set up a community of practice around remote sensing, which is fantastic, and also has created some online guidelines to enhance the uptake of remote sensing for weed detection as well. So Jane's going to talk about the opportunities and limitations of new remote sensing and AI tech as well in terms of weed detection and mapping. So over to you, Jane. Thank you.
Thanks, Justin, and thank you everybody for having me. Hopefully you can hear me okay. Great. All right, I'm just going to share my screen. How's that? That sound okay?
That's looking great. Thanks.
Okay. So this project came to its end in July this year. It's been a project that's been going on for a little while. So I'm going to talk about what we did in this project, who was involved, and particularly some of the outcomes that we've been able to achieve with this project, which sort of aligns a little bit with some of the work that Carlos has been doing. So the project sort of stemmed out of a time when we saw that there was some significant gaps in the technology and AI world for weed detection, particularly in heterogeneous landscapes. There's a lot of research that's been done in crops, monocultures, and there were a lot of people working in silos across Australia. And our approach was to try and bring that together in some way to try and facilitate education and to help us learn further about what this tech can do. So we put together a 4-year project that went from 21 to 24 and funded by from DAFF, Department of Agriculture, Forestry and Fisheries. And we were looking at basically investigating 3 modelled weed systems that would enable us to test this technology. So what were the limitations and opportunities of different sensor technologies and different platforms in mixed landscapes or heterogeneous landscapes? So looking at your RGB, your multispectral and hyperspectral sensors on both UAV or drones and satellite platforms. We looked at 3 different weed systems and landscapes to test this, and they differ in their management objectives, but also their growth habit and their location. So hawkweeds were chosen in Kosciuszko National Park, which is a herb, African lovegrass in the Snowy Monaro region, and then bidoobush, which is a shrub on the north and south coasts of New South Wales. Essentially what we did was to find 3 to 4 study sites per species, and we were aiming to target those plants when they were in different phenological stages. So flowering, non-flowering, height off, so we could then identify the perfect time to be able to take the imagery. We took some vegetation assessments by placing different size quadrats around the target plants, a bit like what Carlos just showed. And within those quadrats, we took some information about what was actually there, both on the target species and the non-target species. And this allowed us to create an imagery ground truth of what was actually there and then compare it back to the later predictions. We took some RGB, multispec and hyperspec imagery flown at various heights and therefore pixel sizes across the landscape. And we used UAV and satellite platforms to do that. And so down the bottom there of that slide, you can see a bit of a pipeline which involved imagery processing, development of an orthomosaic of the site, doing some labeling, developing the code and the models, and then validating that model and leading to detections. So some of this work was to develop, try and look at pipelines for different imagery as well. So just a quick summary of what we tested, different cameras across different platforms. And so I put pictures here of the orange and mousy hawkweeds, or African lovegrass, or the bidoobush. So you can see that for all 3 species, we used RGB cameras. We also used multispectral cameras for a mousy hawkweed, but we did need to use New Zealand data for that because there just wasn't the infestation size here in Australia. And both those sorts of hawkweed species are eradication targets in New South Wales. And then we got bidoobush and African lovegrass information from all the different cameras other than your hyperspectral camera. And that was basically due to some issues we had with wind on the day. What we developed In terms of pipelines, well, we're able to see that there is this common pipeline that's going to be needed when collecting data for weed detection, starting from on the ground with your ground truth data, what's really there close to the naked eye, and then your aerial data, right through to pre-processing, getting that imagery ready to put through a labeling software to be able to tell, then train that algorithm what the target species is versus non-target. And Carlos alluded to the importance of people with weed expertise at that point in time. Then we've got the models that are trained to determine what are the most efficient models, the best models to use for particular species, to then evaluate those for their comparison, the accuracy essentially, which would work best, and then be able to predict, make a prediction map essentially of the species in that site. We produced a number of outcomes, and some of the slides I'll show you in a minute will show you the complexity of that. Looking at, for orange hawkweed, we're focusing on deep learning using the YOLO version 5 model, object detection model with our RGB imagery, whereas with Bush, we used a different approach with the UNet segmentation model with multispectral imagery. And then we also had some lovegrass detections using a deep learning CNN model. And then African lovegrass, again, is different because we're using satellite imagery, and that was more of a machine learning approach using multispectral imagery. So we aim to do a lot of different sensors with a couple of different platforms, trying to work out which machine learning algorithms were best. So we worked pretty hard to try and get as many of those as we could. And this is not meant to show you the detail of these slides, but just to show you the complexity of the models that we tested. And you can see here from the hawkweed ones that we looked at both target species, different computer vision techniques were used in the imagery, whether they're bounding boxes or perimeter boxes around the targets. The different drone imagery we use this with the different sensors. So Microsense Alta multispectral camera versus a Sony, two different Sony RGB cameras or a Fujifilm RGB camera, and different pixel sizes for each of these, versus flower, foliage, or flower heads, and the different sites that we used, and the different models that we tested, from which we then selected the most effective model for use with that particular imagery. And we did the same for Bush. You can see here across the south, central and northern coasts. And then for satellite imagery as well on the same sites. And we used 50 centimeter SkySat imagery, which we are able to obtain from one of our partners, which I'll talk about in a minute. And then again, for African lovegrass, a number of different models that we tried different applications with to determine where this plant was. And that was a real hard one because African lovegrass is a grass and looks very similar to the other grasses. So grasses can be a real challenge, particularly that species. And that's our satellite imagery as well. And I'll show you in a moment if you can access, if you're keen and have this expertise. We have a place where all of that has been documented. But the key challenges that we found out of this project were the technology and field challenges. So just understanding how the cameras are put together, how they talk to the UAV, and also in terms of the AI technology as well. We've got field challenges, wind, light, going out to remote locations, and therefore, an update occurring when you're in the middle of trying to collect data. All of these different technological and seasonal challenges that can make collection of this data really difficult, and a lot of planning needs to go ahead before each mission. And you can see there, we're us on the beach, we had issues with sand, and wind coming across the coast, pushing our drones around, which was, when they're quite expensive, is something to consider. Labeling, that was a really big bottleneck for us. Labeling, again, as I said, is the process of getting a weed expert to identify in the imagery where the weeds are, and to be able to annotate those using particular software. And that takes a long time, particularly when you've got a number of weeds across a single image and you have to be very careful to identify correctly which weed is the weed and which is not, because oftentimes you get lookalike species in the same image that can look very alike to our eye. And so we need to be very careful about labeling that correctly, and just having enough instances to label is really important for deep learning models. They need a lot of training data to be able to work effectively. So that was a real bottleneck for us. And then there's data storage and access. Data storage is, some of these are huge data sets. And so that costs money to store. And then to provide access to that, there needs to be a more common way to share that. Alignment of imagery is important in terms of datums. A lot of people use one datum and then you're collecting data with trying to align it with another datum. So being able to keep that in mind is important so that you can get alignment of imagery. And then accurate field validation as well, being able to really report accurately what is being seen on the ground, so that the prediction maps, we can be really sure of what we're seeing in the imagery when we compare them. And again, it's important to document the failures and tech limitations so that we can, avoid losing our resources. And so all of these learnings was clear that, a lot of people were struggling with similar things. And so we developed the Weed Remote Sensing Community of Practice, which is a cross-sectoral community of people working in this space, from government to universities to land care to students. And it now spans outside Australia to New Zealand, Inner Mongolia, the UK, and New Zealand. Have I mentioned anyone else? Israel, a couple of Israel people too. So we're hoping to grow it further. And what we're aiming to do is just to try and keep bringing together people in this space to learn from each other, to network each other, to provide learning opportunities of what people are doing so they can connect later on, and just to develop and share information and support each other. And that's, I've got the QR code, but I'm sure, you can get my details at the end of this presentation if you'd like to join up. It's free and we just meet quarterly online. And then we developed the Weed Manager's Guide to Remote Detection, which is hosted by our Golbali Institute at CSU. That's a bit of a screenshot there of the opening homepage. And in there, we've got different applications, tabs showing the different aspects of weed detection. And it's more aimed at your intermediate person, maybe someone who's coming in for the first time or has just been working in this for a while. This is a highly technical field, and computer vision particularly is a science in itself. But in terms of documenting the process and the things to be aware of, we've sort of tried to put that together in a way that's user-friendly and therefore links to other people and other information if you want to go further. And so there's menu items like you can see on my screen, things to think about with weeds, but also with data capture platforms, sensors and data analysis. And we've also got some case studies in there of other people and some publications and ways in which to see how all the outcomes from our project, and that's shown in the reports and models page. So we want to thank our many partners that were involved in this. As I said, the DAFF has been wonderful in enabling us to complete this work. We're extremely thankful for their support. But Queensland Unity of Technology, New South Wales Parks and Wildlife, and a number of Shire councils as well, XAG, Illawarra District Weeds Authority, I can see David Pomery's here, and local land services have been really instrumental in helping us at various stages across our program. And here again, here's the link and the website, but you can always just email me and I can give you access to both, or you can search for those as well. I think that's me.
Great, thanks, Jane. And with your permission, we might add a link to your repository in the NRM Knowledge Hub, so the NRMs can hook through to you guys more easily.
Everything's shareable from this project, so more than happy.
That's great, excellent. Thank you very much. Look, we're now going to hear from Andy Sheppard. Andy's currently seconded to DAFF as the co-executive director of the DAFF-CSIRO initiative called Catalysing Australia's Biosecurity. You might have heard of it. He is also the chief research scientist at CSIRO, working on biosecurity and invasive species management We're also joined by Jeff Brosel, the Senior Principal Research Scientist in Biosecurity Research from DAFF, and he's also with the CAB initiative. And Jeff's going to participate in the Q&A session later to broaden our discussion of tech solutions, potentially available in the NRM space. But first, Andy's going to talk about the new technologies that can support surveillance and management of NRM pests, weeds and diseases, and specifically 3 new approaches: next-gen eDNA, genetic biocontrol, and spacecows. So over to you, Andy.
Thanks, Justin. Can you hear me all right?
Yes, we can.
Yeah, so I'm going to get, so thanks very much. Just want to acknowledge my my co-authors, Andrew Hoskins and Kumran Nagalingam, for providing me with the information for the slide. Really, I'm just going to cover two tech opportunities, one in surveillance for management of vertebrate pests, and the other is, which is actually on the ground and running support, and the second one's the genetic control options in not very much detail given the amount of time we've got. Anyway, so in terms of surveillance management for ferals, CSIRO with multiple partners got up a project under the Smart Farming Initiative through DAFF. It's actually a project led by an Indigenous corporation to build a platform to be able to surveil for feral animals for achieving multiple benefits on their land. Lots of partners across Northern Australia, and made quite a bit of media when it pitched itself as spacecows, a landscape scale herd management for unmanaged cattle and buffalo in the Northern Australian Indigenous estates. And as I said, the program is very much run by our Indigenous partners, and CSIRO and other partners like Microsoft provide the technical capacity for that team to build what they're doing. So I'm just going to run very briefly through the project and what it's achieved. It came to an end on the 30th of June, so it's a good time to share what it achieved. So it's really focused on developing a tech-based decision support platform for managing these unmanaged feral herds on country. So it was focused around tech development, both the tech and the data platforms to be able to do the analysis, Indigenous training, particularly around the tech, but also ethical mustering, protection of environmental and cultural values, and to create and support viable on-country business opportunities. But it has a complex context, right? Because these Indigenous corporations are running pastoral and buffalo enterprises. So they're providing into pet meat, the live trade, safari hunting, and crocodile food. So they've got multiple businesses. There's also the importance of these animals for local meat consumption, impact on traditional natural resources like crocs, turtles, and water lilies, impact on environmental values, impact on cultural sites as well, and carbon benefits. So the whole point of the project was to be able to try and capture and support all of these benefits around. So where did we do it? It was done in remote parts of far north Queensland, Cape York, and also on Arnhem Land. So it was innately tough and difficult territory to get out to. Why was it done? Because currently buffalo populations are having a huge impact on northern floodplain wetlands, as you can see from this. So it's around how do we better manage feral animals at a landscape scale to be able to manage for these kind of impacts. And it was built on, as I say, developing a tracking technology platform that would be able to monitor and understand where these feral herds were in the landscape and provide information that would support management decisions. To create effectively the world's largest remote herd monitoring program. And it was built on satellite tracking of using ear tags that were sourced, from off the shelf from the marketplace, but a long process of getting those ear tags out onto the animals and being able to monitor, obviously, a proportion of them in the landscape to be able to understand and predict where the herds were there. So it had quite a complex plan to address all these challenges. So as it was set up, the Smart Farm project was around defining an operating area, making sure that we have a mustering method to be able to be informed by the technology, identifying, developing, and installing the ear tags, having the monitoring and digital platforms to be able to collect and analyze the data, working on targeting particular herds and animals, having to deal with the handling of those animals in order to be able to provide, install the ear tags and releasing them, and also recognizing that there is tag loss and need for tag retrieval. And then also in terms of getting the satellite tagging system to work, it was around making sure the technology worked, activating the tags, putting them in place on the animals, getting those tags to download data effectively through the satellite and on-ground data tracking system, and getting that data back to base and data visualized to provide the information you need. So what kind of movement field data was collected? Well, there were animal density surveys done through helicopters to support the satellite tracking. The satellite tracking itself tracked the movements of the animals across the property in the context of habitat suitability. There was also the tracking of the management activity. So we tracked the vehicles as well that went out and understood the difficulty of terrain they were moving through and how efficiently they could get to particular parts of the property to undertake the management. And from the data was able to forecast herd movements and where herds were in the property for future harvesting decision making through a data analytics platform. The main output from the project is a number of business-specific dashboards that are available and can be used now in real time by the businesses on those properties to be able to manage feral animals. These dashboards provide the forecasts of the herds in real time as to where they are on the property. They also link to meat prices, so the owners can understand where the meat prices are optimal in terms of being able to harvest them to get it into the meat markets. Includes also fuel prices, so you can understand the costs of getting out to these regions to be able to undertake the management. They inform managing efficiency, particularly in the remoteness of these locations, and the viability of harvesting. They also provide you measurement of the carbon benefits based on the number of animals that you've harvested, and measurements also on environmental and cultural benefits that you've achieved through these landscape activities. And now the plan is to scope it out and take the platform to a whole range of vertebrate pests across the north. And here's the first view of how we want to take space cows as a broader feral animal survey to be able to understand and manage where these feral animals are at a much greater scale, and be able to look at Northern Australian scale carbon impacts of harvesting to much better manage our feral animals into the future. Then I'll just talk, second part of my talk's really about some of these genetic control approaches. As I say, I haven't got time to go into great detail, but I'm going to primarily tell you about the two main approaches that are being developed. The first one's called RNA interference, or RNAi, which is a topical way of applying a solution containing double-stranded RNA sequences that switch off genes in your target organism by mediating growth and reproduction. And then secondly, I'll come on to the more widely known and more controversial gene drive systems. So again, I don't have time to go into this in detail, but this gives you basically a schema of how the development of this topical or spray-on RNAi is developed, but it's basically identifying target genes and then developing sequenced RNAi that will can be introduced when they get into a cell, can be introduced and will attach to and switch off key genes. So it's exogenous gene silencing as a way to reduce the fitness and improve the management of some of these pests and diseases. I'm going to talk specifically about some of the work that's actually happening on weeds. I'm actually today at a workshop here in Brisbane with some of our American colleagues around how we can progress RNA management for weeds, but there's a commercial product available in the US for southern corn rootworm, and some very important work's been done by Nina Mitter at University of Queensland, applying this for the management of viral diseases of plants. So it's a technology that's very much coming onto the market, but from a weeds perspective is still under development. So the Americans have been working on RNAi to control Phragmites australis or common reed in the Great Lakes, but through this gene silencing approaches, and they've been looking at genes that to target growth, seed production, vegetative reproduction, photosynthesis, reproductive organ development, and pollen production. But the work we've been doing in Australia has been focused on a much more important weed for us, which is wild radish. And it's one thing to be able to develop your RNAi active ingredient, but you need an effective formulation to be able to deliver that in a sustainable manner into the field where it can persist and continue to be active, which is a lot of work where the UQ group has been doing. But we're at the process where we've just managed to produce proof of concept that we can suppress and knock down growth in wild radish in a laboratory setting. And we're now looking at how we improve the delivery to target cells through the plant epidermis, and also understanding the costs of these kind of herbicides and their scalability. But because this is going commercial, they've now got we know already they've got the commercial viability of RNAi down to about $1 a gram, which is a huge amount of active ingredients. So it's becoming very commercially viable. Second system I just want to talk about briefly is gene drives, which I'm sure many of you have heard about. Even the minister last week, Minister Plibersek, talked about the development of genetic opportunities to control feral cats. So I just want to give you a bit of background. Again, here's the schema for how you build a gene drive based process, which is basically embedding through genetic manipulation genome sequences into your target organism that will reduce the fitness of that organism. So it's a GMO, as having control systems so that the gene drive can perpetuate and suppress populations at a regional or a local or regional scale. The first successful development of a gene drive system was in the malaria mosquito. Again, driving these mosquitoes to a single genotype, which has a much reduced capacity to transmit malaria. And that's a program supported by the Gates Foundation called Target Malaria that's operating in Africa, and the approach is spreading around the world. But where it's been developed primarily in Australia is for control of rodents. This is a diagram of what a myonic gene drive looks like. You've got your gene drive mouse, and every wild type mouse that any of these gene drive mice mate with, all of the offspring are male. And there are two approaches. One is you can do it all synthetically using CRISPR-Cas9 gene editing, but the approach that's been developed in Australia is actually built on an existing selfish sex gene in mice called T3, where we're trying to augment the presence of that selfish gene in the system. Again, we got through the collaborations, this is work by Paul Thomas at University of Adelaide. We now have proof of concept in the lab for a working gene drive system in a mammal. That's the first time in the world where that's been achieved. So it's a great achievement, and it's an Australian achievement. And Macquarie Uni have also developed a system now that is proof of concept in another vertebrate, which is fish, with the aim of coming up with a solution that may be able to help us long-term in suppressing our feral carp populations. In order to do this kind of work, it's complex, and it requires a lot of interdisciplinary work, a lot of social science. And so we have a big international consortium that's been working together to develop it. We're in the very early stages, and again, the workshop I'm at today is discussing it, of developing gene drives in plants. Plants are much more complicated. They have very complicated reproductive systems, but the thinking and the research is starting. There's actually more work going on in the US than there is in Australia at the moment, but weeds are another option that we're working on. But these kind of technologies are controversial. They're GM, and you need, before you even start, to ensure there's a reasonable amount of social acceptability around the science itself and the risks. And as part of our consortium, and in Australia, we've done a huge amount of work to look at social acceptability of gene drives, particularly in feral cats. And we've published a report on this, and the public acceptability in Australia is pretty high. But you tend to get two responses from the community. One is they think it's amazing and we should do it. We should have done it yesterday, and others that get really concerned about the control of it. So you have these extremes in terms of views that you have to manage. And in order to address that through the Science Journal, the consortium produced a principles of gene drive research around how we will do it in a transparent and accountable way to ensure that the public understands exactly what we're doing. Both of these technologies have a pathway to deployment here, from conceptualize right the way through to regulatory approval for deployment. This is a very long process, particularly the regulatory approval. Nobody's released a GMO for an environmental benefit. So there's a long process, particularly for gene drives. And these two technologies are different stages through this pathway to deployment. So to summarize, there are plenty of technologies out there that have real potential to help us manage feral pests and weeds. We can't ignore them. We should test them in terms of our context. So our role is as science agencies is to understand and regulatory organizations to understand the possibilities and let society ultimately decide if we want to use it. And that way we can ensure that we can adopt these technologies using a responsible innovation approach. Thank you. Thanks, Justin.
Thanks, Andy. That's fantastic. And it was a bit of speed dating in presentations, but really interesting and exciting stuff. So lastly, before we head into our Q&A session, we have Susan Campbell. Susan is a research scientist with the Invasive Species and Environment Biosecurity branch of DPIRD WA. And for the last 15 years, Susan has explored the innovative surveillance and control solutions that are available for invasive pest species, and particularly specialising in automated acoustic detection of pest birds. Her expertise also extends to the management and control of other vertebrate pest species, including rabbits and foxes. But today, Susan's going to talk about her experience in using acoustics integrated with machine learning to detect pest birds. So over to you, Susan. Thank you.
Thanks, Justin. I think I got that sorted. Can you see my full screen now?
Yes, it's all good. Thank you.
Yep, great. All right. So as you've just heard, being able to deliver innovative technology to enhance biosecurity outcomes still does take a lot of work. It's just a different kind of work. So hence my play on words for my talk title today. And several of us have been talking about how we incorporate AI and machine learning into our biosecurity responses. So I just wanted to be very upfront and share with you that the motivation for exploring these AI and machine learning tools at DPIRD in Western Australia, it does not come from a desire to reduce or replace humans from the very complex process that is biosecurity responses. And I'm sure I'm not alone in that sentiment. It's across the board. And rather, as researchers, what we're trying to do is to bring these AI to our projects so that we can arm our staff with more powerful and more speedier tools that enhance their critical subject matter expertise. So that idea of being able to speed up and have that real-time data available is critical for our managers in our line of work, because as we know, being able to detect and respond to incursions early is absolutely paramount if we're going to mount a meaningful response. But unfortunately, this task of trying to detect pests early is becoming both increasingly demanding with the exponential increase in global trade and tourism. And it's also becoming a lot more complex with the myriad of ways that pests cross our borders, both deliberately and accidentally. And this chart here just gives you a snapshot of how substantially biosecurity responses that are managed by DPIRD just in West Australia alone have increased substantially over the last decade. What it also shows you is that out of all of these responses, only 2 of them are vertebrate pest animals. The last line of green that you can see there, sparrows and starlings. So both of them pest birds. West Australia takes pest bird biosecurity quite seriously, and the reason for this is that when it comes to pest birds, West Australia still manages to maintain quite an enviable biosecurity status. We remain free from established populations of the majority of introduced pest birds that tend to plague the eastern seaboard and beyond. And by and large, the responses that DPIRD leads in the bird space are predominantly geared towards eradication or area freedom. And from my perspective, this presents a raft of innovative research opportunities. And my role for the last decade or so, as Justin mentioned, has been looking at how we can incorporate acoustic surveillance into WA's long-running starling control program. So why starlings? They're a high priority declared pest for West Australia. They've been subject to ongoing control since the 70s. This little video here is a wonderfully hypnotic scene of a murmuration of starlings from over in Europe. And unfortunately, thanks to our friends from the Acclimatisation Society back in the late 1800s, bringing them across to Australia, they're now firmly established throughout the eastern seaboard, Tasmania, New Zealand, and even further beyond. But they're not established in WA, and we would like to keep it that way. There's a long list of damages directly attributable to starlings. They destroy crops, they foul stock feed, they transmit disease, aggressively outcompete our native species for hollows. They'll spread weeds, and there's also a number of undesirable social ramifications like fouling of infrastructure. And these couple of short videos here are just a nice visual, timely reminder of a couple of the reasons we'd like to keep starlings out of Western Australia. Unfortunately, a lot of West Australia is suitable habitat for starlings, and there's every chance our landscapes could support over 12.5 million starlings quite easily. So at that point, we would be incurring losses in excess of $180 million to agriculture. Our control program has seen over 50 years of near continuous management. It currently operates over a very large area, which I'll show you shortly, using a combination of predominantly live lure bird trapping, also shooting at times, and netting when it's appropriate. There's two focal areas for the control program. One is at the South Australia-West Australian border, either side of Eucla, and then down on the South Coast region, either side of Esperance, another large area where trapping occurs, sometimes extending as far west as my hometown of Albany, down on the southernmost point of WA. This chart shows the number of starlings that have been removed from those two different areas. So the border region in red and that Esperance South Coast region in blue. Unfortunately, investment has varied substantially over time. So these charts not corrected for control effort. Back in the mid-2000s, there was a high level of investment in removing this incursion on the south coast of WA, the Big Blue Peak, which was highly successful. And for several years after, we had zero detections of starlings on the south coast. But that outcome is confounded by the fact that funding for the program was also reduced quite substantially over those periods. So were we not catching any starlings because there weren't any there, or were we not catching any because we weren't looking hard enough? So that's where acoustics enters the program, with an opportunity to be able to undertake remote 24/7 surveillance over a much larger footprint. The concept of acoustic surveillance isn't new. There are many different commercial types of recorders on the market. They can operate underwater, in deserts, really any environment where there is a benefit to monitoring an audible species. But the one significant drawback for the units that you can see up on screen is that they all record their data on board, typically on memory cards. So this isn't ideal for a few different reasons, particularly in the biosecurity space. Firstly, field sites can be a really long way away and difficult to access. Secondly, it takes a really long time to download and manually listen to what can often be multiple gigabytes of acoustic information. But most importantly, from an early detection perspective, if you're looking for a pest, you may not receive the notification or find out that that pest was in your area until several weeks, typically months after the event has occurred. So the device here is different. This is an edge computing acoustic surveillance device, and thanks largely to support from the Centre of Invasive Species Solutions, we've now constructed 4 of these proof of concept towers, which are providing us with end-to-end communication and detection of starling incursions on the south coast of Western Australia. So real-time detection is now possible because the audio is processed on board the devices themselves. This occurs on a Raspberry Pi compute module 4 inside the box. And being able to run the algorithm on board the field device means that only small packets of data need to be remotely communicated. Currently, we're using Telstra's low bandwidth network for that communication service. Which does mean that our towers currently need to be placed in an area that's got a bare minimum of Telstra coverage. But obviously, given the improvement in accessibility of satellite communications, we should be able to move away from relying on Telstra in the near future. But we do have this real-time communication, which is terrific. I've conducted some playback experiments at the units to determine their footprint, what their surveillance footprint size is. The microphones themselves are terrific. They will pick up realistic playback calls from over 150 meters away, but at that distance, the signal to noise ratio is a little bit faint for the algorithm to work reliably. So realistically, the automated footprint of these units is around 60 to 80 meters, but very variable depending on weather and terrain. The algorithm itself for starlings, it was designed using a convolutional neural network machine learning algorithm, and it's working really well. After several training iterations, we've got really pleasing accuracy, precision, and recall rates. It was trained to pick up the 2 types of calls that you can see up on the screen, a couple of variations on what we called a buzz call, and then a very shrill descending whistle. Starlings are incredible mimics, so a terrible choice of study species when you're setting out to try and automate their detection, but fortunately they did tend to incorporate these couple of signatures fairly reliably into their repertoires. At each step in the model's evolution, the algorithm's being trained with calls collected predominantly from South Australia because they have lots of starlings and we don't. And on board the device, this is a very broad overview of the process that's occurring. So very short snippets of audio are queued for running through that CNN algorithm. And then those short 5-second snippets, which contain the putative starling detections based on the confidence level that we've set for the algorithm, are then also queued before sending to a cloud server. We have written a few other scripts which enable us to train or retrain that algorithm and also enable us to communicate remotely with the field devices themselves, so we can push up firmware upgrades, for example. And another terrific output from this work has been taking the algorithm that we built for the devices and being able to run it on a desktop computer. It's allowed us to process archived acoustic data, and it's also been licensed for free by the sponsors to share with conservation agencies. So we've taught the algorithm to forget starlings and to learn other species like western ground parrot here in WA, for example, to help with the detection of threatened species as well. And the last piece in the puzzle was to design a user interface so that we can view, playback, so listen to, share, map, verify the detections that are coming in from those field devices. So each detection gets its own line in this interface with a little thumbnail. So once you have your eye in, you can very quickly verify as starling or not starling just based on the sonogram, or you can dive in and have a listen to the playback as well. So this interface has helped me to verify the detection footprint of the units, but more importantly, it's also helped satisfy our field staff, our biosecurity officers, who at the outset had a very healthy level of reservations around the use of this technology. And unbeknownst to me, earlier in the year, they placed a starling lure cage with live starlings directly under one of my towers. And my very excited calls back to them in the following day or two were met with some pretty wry grins and knowing smiles. So it's always nice to have that win when the science did come through, eventually proving itself after what's been quite a long R&D journey. And just moving forward, over the next 12 months, I hope to roll out 15 to 20 more of these surveillance towers for starlings. This time they're going to be on a more portable tripod solution so that we can be even more adaptable in where we choose to roll out that additional surveillance. So we're working well to transfer this technology and incorporate it into the starling program, but we're also progressing it across onto other species as well. We've got about 100 sulphur-crested cockatoos estimated to be in the southern part of Western Australia, where it's not their natural range. So we're now bringing acoustic surveillance on board to help our response in that space as well. Thank you.
Thanks, Susan. That's fantastic, and thanks to all of our presenters for generously giving up their time and their expert knowledge today on the things that they've been working on and using to improve the way we work. And I think Susan made a really key point in that this is not about replacing humans, but I think the opportunity to reallocate human resources to where they can be most effective is a really great opportunity for the NRM sector. So we have about 15 minutes or so for a Q&A. We have had a few questions come through the chat area, but if you do want to ask a question now in person, I'd ask you to come onto camera and place your hand up, and we can do that as well. But whilst people are thinking about what they might like to ask in around how these different sorts of texts might be able to be used, I thought I might just pick up a few things out of the chat. Carlos, thank you. You've answered a whole pile of things, so thanks for those. Just a few other ones have come a little bit more lately. Andy, I think to you, the use of the topical application of RNAi, is there potential for using that with arum lily?
To be honest, we actually don't know enough about which kind of plants are going to be most susceptible.
Mm-hmm.
You know, the Americans have been working on Phragmites. It's turned out to be a real challenge for them. You know, it all depends on how tough the epidermis is and how easy it is to get the topical RNA into the cells, right? So we actually don't have enough to say, but at the moment, you know, we're trying to understand what kind of aspects. You know, we're wondering whether water weeds might be a bit more susceptible because they tend to have thinner epidermises. So, you know, but potentially anything at the moment, we just don't know.
Yeah, that's great. And sort of similar question. I know that the gene drive work on rabbits, on mice is predicated on, you know, highly fecund, shorter lifespan animals. What do you think the potential might be for aquatic animals like cane toads? You spoke about carp before, but no, what about cane toads?
Well, cane toads are obviously a target that's been pretty high on our priority list to try and think about. And you know, there are multiple ways that you can manipulate using genetics animals so that they so that you can suppress their impacts. And the approach that Rick Shine at Macquarie Uni with the genetic colleagues are taking with cane toads is actually preventing metamorphosis, because it turns out that the biggest predator of cane toad tadpoles in the breeding areas is other cane toad tadpoles. So if you can stop metamorphosis, the tadpoles will eat each other and then not be able to, you just get one very large tadpole at the end, which can't metamorphose into an adult. It's a bit weird, but so that's the approach that they're taking at the moment, because we know a bit about cane toad metamorphosis. So there's, for every particular target, there could be a unique set of solutions, and that's currently the most promising one for cane toads.
That's great. I'm waiting to see some questions, some hands pop up, and I'll keep on pinging questions at you. And this may be just a question for everyone on the panel who presented today. What do you see as both the opportunities and the challenges for us getting this tech into applied use within the NRM sector in terms of people working on endemic pest, weeds and diseases? What's if there's a key takeaway for us in that space, what do we need for that? Is it networks? Is it connection? Is it funding? What's the thing that really we can do to help you guys and you can do to help us? So, Carlos, I see you've come off mute. Would you like to respond?
Yeah. I suppose funding is a very challenging factor always, especially when it comes to endemic species. But the efficiency or the efficacy of that funding. So probably you mentioned networking and sharing knowledge and building solutions that can be reused and easily taken up by others. So like in computer science, it'll be sort of like the open source space. It has revolutionized many other industries, and in fact, AI itself has come off the back of open source software and open source processes. So that sharing the knowledge and sharing processes that can be taken up by others and working together, collaboration, that thing that will make the best use of funding.
Thanks, Carlos. And listening to your presentation and also Jane's, it made me wonder about whether there is a role for the NRM sector in helping with some of that, the training of the AI models, some of the field validation work, and wondering whether there is an opportunity for us to assist researchers in that space more than what's been done already. So if that is useful to you in the sort of work that you're doing, then we're really happy to help facilitate that. So
absolutely, it will be of great value. Amazing. It's an amazing opportunity to probably collate a national database of weeds, national database of weeds or other species that can be worked on by hundreds or thousands of practitioners across the country. That would be an amazing outcome, absolutely. And again, it would have to be in somewhat of an open source environment.
Right. Jane?
Yes, I think that's exactly right. What I agree with Carlos is that we need a national platform where data can be housed and shared in an equitable, fair way, which is probably to a set of standards so that it can be run through the same software. And I think that's probably what you meant, open source or cloud-based, or and that's something that our team have been talking about with Australian Scalable Drone Cloud guys, who was essentially starting to try and build that work, that framework to be able to put drone plant information into the cloud. It gets processed and spit out the other end. And some of that will be a fee payment process, I'd imagine, but mostly to be able to be sharing that in a central location in a standard way, I think would be valuable.
Yeah, that's great. Excellent. Susan, your technology around acoustics, is there potential in the future for that to be used to determine presence, absence of established pests for better planning for where works need to be done?
Oh, absolutely. Yeah, for sure. Anything that makes a moderately unique acoustic sound that can be separated out from the background noise. One of the fortunate things about where I've tried to test this technology at the moment is that there's not a lot of industrial background noise. So when we talk about rolling these devices out at ports, for example, although that's an early incursion example again, there'll be additional challenges in terms of machinery noise and just getting that good quality signal to noise ratio. But in terms of established pests, if they make a noise, for sure, you can determine presence, absence.
Yeah. And how much is Starlink? So how long is it going to take before we get sort of tech, do you think, able to be used in like an NRM sector, so less of a research space? And how much is things like Starlink going to be a game changer for that?
Starlink as opposed to Starlink.
Oh, sorry. Starlink.
Yeah, no, for sure. I think our next rollout of these devices will certainly lean upon the Starlink technology and the coverage that can provide. The nearest, to be honest, communications, even on the remote south coast of WA, have not been insurmountable. By the time you put something up on the end of a very high pole and give it a directional antenna, it increases the expense. But our nearest Telstra towers to any of those devices that you saw are well over 20 k's away. So yeah, it's not insurmountable. The cost of what I showed today is probably a barrier for the NRM space at the moment, I would say. But as with any of the tech that's being presented, that will change rapidly. I imagine that, well, I know that there are people looking to commercialize the package that I've delivered to you all today. So hopefully that will only just, see the costs come down and become more affordable.
And this is just a question from Karen Maloney. Can the device do multiple species at once, or is it limited just to a single species at any one time?
No, for sure. The computing power that's on board at the moment would certainly be able to handle running multiple algorithms for different species. We've not done that yet, but it's well within its capabilities.
Yeah, great. Excellent. And Carlos, a question for you, which I'll paraphrase out of the chat, but from Lachlan Fittler. Do you have any biosecurity officers currently using UAS to capture this style of data? Flying these missions and processing can be a specialised skill set requiring significant training and licensing. This is for drones. So, and have you had any success incorporating drone work into biosecurity operations just more generally? So that whole issue of drone operation seems to be a challenge. Is there any reflections you've got in that space?
Absolutely, yeah. It has been a challenge, and for the last few years, to get someone trained, it takes up to 5 days for using a big drone, and the cost of that, plus the cost of licensing, plus all the operational challenges that come with it are very big. But in saying that, there's also many advantages that are coming along. So our chief remote pilot has set up a policy that has been approved to fly micro drones with much more flexibility. So you still have to do a certain course or approval by CASA, but it's much more streamlined. And we have been trying detections and even live detections from the live feed of the drones on those mini drones, and the results have been promising. So yeah, you can still I think we're moving in the right direction, and technology is advancing very fast, and we are doing the groundwork to catch up with that. So I think in the future is going to be longer flying drones and much more processing power out in the field. So yeah, the future is looking bright. We need to keep on catching up with it.
Yeah, it sounds like some of the work that Susan's doing in terms of onboard processing with the work you guys are doing, it sounds like some interesting crossover. Jane, this issue of complexity and operation and what have you, is this something you've discussed in the community of practice for the remote sensing area, and can people draw upon some chats or information there?
Absolutely. It's the thing that comes back is, okay, we can go out and get some drone data. Now, what do we do with it? How do we make this useful? And this is where the issue lies, is that computer vision, the AI side, is highly specialized. We can't just go out and do a 3-day course and learn it. We need access to people who can do that. And that's, I think, where Tim, Australian Scalable Drone Cloud, was trying to take out some of that to do that all in the back end. And so then you could, if that sort of a framework, I think, would be something valuable, because then, you know, it takes out that complexity, and allows us to just upload the data and get something out at the end. I think that's the holy grail.
Great. We're probably coming close to the end of our time. I've got one more question of my own, but I'll just wait and see if anyone pops anything in the chat in the next 30, let's say 5 seconds. So I'll pad for a moment. That's not quite 5, but I'll go with this. Andy, my last question's for you. It's around the spacecows project. Sounds like there's some really great data in there to better plan on-ground projects in terms of understanding where animals are and they're not, similar to the song meter work. Is there potential application for that at the pre-project phase to better refine where works are going to be done, and what the outcomes are likely to be, and, you know, some of the economic data that you're collecting around travelling and vehicles and the like?
Well, look, so I think, I mean, I think the whole purpose of the platform is to be able to provide mustering advice to these corporations to be able to optimize the benefits they get and the economic return they get on their mustering activities. So the platform, I mean, the platform is still, you know, it's available. It's not scaled up yet, right? It's a research proof of concept, but certainly the community that we worked with is incredibly enthusiastic about incorporating, or the communities, I should say, incorporating into their business model. I think the way this flows into broader benefits is, on the one hand, that, you know, that the demonstrative value of it is recognized and promulgated through the local communities. I mean, there's nothing, this technology works equally well for extensive agriculture, livestock agriculture as well. So it's all about getting the word out there and understanding how we can use satellite tracking much more effectively to support decision making. And then broadly, you know, more broadly, we can move into managing feral animals more collectively across the north. Instead of punctuated programs, if there's you know, all the evidence suggests you can get really good carbon benefits from effective feral animal management, that you can, just like the Northern Australian traditional burning program, you can generate carbon credits that attract big corporations to come in and invest. So I think it's got a good track way to adoption, but we just need, you know, there are many hurdles still, and we need to make sure that it's valued by the current beneficiaries, and that we can promulgate it. And we'll probably need a bit of support to be able to promulgate it more broadly. So.
Well, I think thanks, Andy, and I think from the chats and the responses we've had so far, it's a lot of excitement in terms of trying to work out where we can apply these technologies into the NRM space and elsewhere. So I think through our involvement with the CAB, I think we can help build some of those connections, and hopefully we've created a few others today. So thanks, everyone.
That's good, Justin. Yeah, I mean, Catalysing Australia's biosecurity is all about supporting the development of these platforms and getting them wide applications. So, you know, the great technologies we've heard about today, how do we make them available for the next problem, you know, rather than having to build it the ground up for each individual problem we're trying to target.
Yeah, absolutely. Thank you. All right, look, I'm just going to close out in the last few minutes we have. So thanks again. If you can show your appreciation through the reactions, if you can offer some claps or whatever, that would be fantastic. Thank you very much for everyone who's given up some time today to present. Our next webinar topic is going to be on the 26th of November, and it is focusing on dingoes as a landscape process. So we're going to have a number of presenters explain to us where dingoes fit into the landscape and the various roles that they have in those landscapes. So that will be on the 26th of November. So if you're in the NRM space, keep an eye on the Slack channel, and for others outside, we'll send through some updates on that for you. Also, for the NRM people that are on board, we have an environmental biosecurity workshop on the 16th of October from 1 to 2:30. That's going to be exploring some of the capabilities and capacities that sit in the NRM space related to environmental biosecurity, in terms of things that you've done, things that you need us help with, and things that we can help promote for you and link you to in terms of environmental biosecurity management. So please keep your eye again on the Slack channel and emails for more details on that. But we're hoping to get some more of the NRM practitioners on board to explore some of those topics. We have, for our environmental community of practice, we have a meeting of that, which is on the 12th of November from 1 to 2:30. So just a reminder that the next COP meeting is then. And also, last but not least, in terms of things that are coming up, we will have our next newsletter, Managing Biosecurity Better, which will be coming out in the beginning of November. For the NRMers on board, that will most likely go to your CEOs. So it'll also be promoted through the Slack channel, so you will see it. But if you haven't got it, go and see your CEO and say, hey, did you get this email with this, and can you send it on to me, please? So yeah, just a reminder on that. Lastly, before we let you all go, we do have a very short Menti survey, which Sarge is putting on screen with the QR code. It's really quick and easy. It's really just for you to give us some feedback on things that we can improve on, things that you liked, any ideas for future webinars and the like. So that is your opportunity to give us some quick feedback. It should only take you 2 or 3 minutes. The Menti platform is really, really quick and easy to provide feedback. So I really appreciate if you could spend a moment to scan the QR code and jump on that. That would be super. And look, lastly, can I just say again, thank you to all of the presenters and the attendees. We're really grateful for everyone's attendance. If you couldn't scan that QR code, there's also a link in the chat. Thanks, Sarge, for that. So, and look, thanks also to Sarge and Trish for all of the support for today. So, really appreciate everyone's time. And with that, we'll say goodbye and enjoy the rest of your days.